All right, this is chapters 20 and 21. We'll be looking at the blood and the heart. So blood is the last connective tissue we're going to discuss. This is fluid connective tissue, and all the components of this tissue is suspended in fluid. So if we had a vial of blood, right, in here are cells, right, and then dissolved solutions, ions, proteins, nutrients, all kind of dissolved in a water base. Now, if we are to spin this vial down, right, this is called centrifugation. By spinning it down, it collects all the heavier particles that are in suspension, and they begin to fall out of solution. We call this precipitation. You allow that to happen over a longer period of time, and you end up with solids at the bottom with fluid at the top. We call this fluid plasma, and we call the solids the formed elements. Now this plasma has inside of it all of these dissolved nutrients, so ions, proteins, even proteins involved with clotting, as well as any nutrients it needs. The formed elements are composed entirely of cells. So how much plasma, how much formed element we have can vary, but typically about 55 or so percent formed elements, 45 to 55% plasma, depending on our blood count. I want to now focus on the formed elements. So the stem cell for blood has a couple different names. I use the term hemocytoblast. You can also say this is a hemopoietic stem cell. This is going to create two cell lines. One is the myeloid stem cell line, and the other one is the lymphoid stem cell line. Now, the lymphoid stem cell line just creates one type of cell. This is called the lymphocyte. The lymphocyte is one of our white blood cells, which we call leukocytes. The myeloid stem cell creates everything else. So, it can create the other four leukocytes. They are neutrophils, monocytes, eosinophil, and basophil. And there are different cell lines that do this. You can ignore all this extra stuff. I want you to understand the stem cell, the hemocytoblast, also called hemopoietic stem cell. It breaks up into two stem cell lines, the myeloid stem cell line and the lymphoid stem cell line, and then cut through all of these breakdowns, and I just want you to understand the key component. Lymphocytes for lymphoid line and everything else in the myeloid line. It's also going to create a cell called a megakaryocyte, which is going to form the platelets. And finally, it's going to also form the red blood cells called the erythrocytes. Those are our red blood cells. Let's look at the red blood cell. Now, the erythrocytes, the red blood cell, makes up the majority of the formed elements. In fact, it's about 99.9% of all the solids. That's because the major role of blood is for oxygen transport. And there is a protein packed in the erythrocytes called hemoglobin, which binds oxygen. In fact, the cell has removed the nucleus. That's why the cell has this biconcave appearance. These cells are considered terminally differentiated. They're not going to last very long. They're made in mass production. Most organelles have been removed, and they're just filled with hemoglobin, and hemoglobin can bind oxygen. Now, you may have heard of the A-B-O blood typing. Right? Someone may have A blood or B blood. They might be AB blood or O. These blood typing are based off of a protein with a sugar group on the cell surface, and we call these antigens. An antigen is any biological molecule that is recognized by antibodies. Antibodies are large protein molecules created by our immune system, and their job is to detect foreign antigens as opposed to self-antigens. So we can break up antigens as self versus non-self. Okay. The whole function of antigens is to make sure, or the whole function of antibodies is to scour the bloodstream and scour through the body to find anything that doesn't belong. So if you are an A blood, you have A antigen. If you're B blood, you have the B antigen. If you're A and B, you have both A antigen and B antigen. And if you're O, you have no antigen. What that means is your blood plasma will carry inside of it antibodies against the other antigen. So for instance, if you're type A blood, your cell, that is your red blood cell, expresses the A antigen, and you're going to have antibodies against B blood. So we call those anti-B antibodies. If you have B blood, then your cell expresses the B antigen, and you're going to have anti-A antibodies. If you have AB blood, your cell expresses both A and the B antigens, and you're not going to carry any antibodies. This is why they say that AB blood is the universal acceptor. And if you're O, you don't carry any antigens, but you'll have both antibodies. This is why they say O blood is the universal donor, because if you remove the formed elements, the cells, from the plasma, you can then resuspend these cells in a nice buffered physiological condition solution and donate this blood. Because this blood does not carry any antigens, therefore antibodies that other people are carrying or not carrying can't attach to them. Now, if you end up having an antibiotic reaction, it's because you have antibodies expressed in your blood that is activating or is binding to antigens that that antibody is against. And this can cause clumping of blood cells called agglutination and ultimate breaking down. So when you break open the cell, it's called lysis. If it happens to a blood cell, we call it hemolysis. And this would happen if you gave A blood to someone who had a B blood type. The A blood cells would react to the anti-A antibodies, and that would cause a problem. Okay? We will talk about the cells that create the antibodies when we get into the immune system in Unit 4. But I just wanted to introduce the idea of antigens versus antibodies since we're dealing with blood. Okay, that is erythrocytes. The next cell is called the megakaryocyte. These cells create the platelets. Platelets are not cells, they're fragments of cells. So what happens is, and the mechanism isn't fully understood, as a cell is expanding, it begins to tear off pieces of itself, and these little remnants now are involved with blood clotting. It's the platelets along with other proteins found in the blood that will be activated to cause the clot to occur. And that's why this is still considered fluid connective tissue, is the protein fibers only come together during a blood clot. And so you become a very solid tissue in a very localized wound area. So both the platelets and some of the proteins will then come out of solution and cause clotting to occur. Finally, we have our white blood cells. These are referred to as the leukocytes. And we can distinguish leukocytes based off of two things: the nuclei shape and cytoplasmic staining. We can classify the different leukocytes as a granulocyte or an agranulocyte. So granulocyte means the presence of a grainy cytoplasm. I'll just put little quotes in there. And this is more, agranulocytes, more of a clear cytoplasm, not as grainy. Okay. So for instance, the lymphocytes and monocytes are considered agranulocytes because of the way the cytoplasm looks. It's very light. The neutrophils, eosinophils, and basophils, even though the neutrophil isn't really that dark stained, it's considered a lighter stain. And then you have medium staining and very heavy staining in the cytoplasm. You can also look at the differences by the shape of the nuclei. So for instance, neutrophils have 3 to 5 individual lobes, but a very light stain cytoplasm. Eosinophils usually are bilobed. There might be 2 or 3 lobes of the nuclei, but they have a very clear, distinct, pinkish, granular stain in the cytoplasm. And these granules are secreted via exocytosis and release things like histamines or antihistamines that work with inflammatory responses and what have you. So it just depends on what their functions are based off what they're secreting. The basophil has also a couple lobes, but it is so dark staining that it's very obvious to make out. And I would recommend that you look at the blood cell demo for the histology section and get a feeling for what these cells look like. Now the monocyte has more of a kidney bean or some type of indented nuclear structure, where the lymphocyte is nice and spherical nuclei. Right? Now we can, there's a mnemonic to keep track of the abundancy of these cells. Remember, these cells make up less than 0.01%, or less than 0.1%, sorry about that. Less than 0.1% of the formed element, so not a lot. Okay. So the mnemonic is never let monkeys eat bananas? So never, neutrophil, let L, lymphocyte, M, monkeys, monocyte, E for eosinophil, and B for basophil. And these are roughly the numbers they have. So in general, of the 0.1%, about 60% of them are neutrophils. About 30% are lymphocytes. Okay. Neutrophils are kind of our first responders. So we want to have them in abundancy. And lymphocytes is the majority of our immune response. So we want them to be in abundance. The rest of them will only proliferate and grow based off of allergies and infections we might have. So in third place is monocytes, about 3 to 10%, and then super low are eosinophils, and the lowest is our basophils. So this is less than 1% of 0.1%, or this is 60% of 0.1%. So it's not very, very abundant. They will expand and grow when necessary. So here are some of the functions that they do. So for instance, eosinophils and basophils are responsible for allergy and inflammation. Neutrophils, as I said before, are our first responders. They have the ability to undergo phagocytosis, right? If you recall phagocytosis from your general biology class, that's the cell engulfment, cell eating. The monocytes play a role with macrophages. Macrophages are also phagocytes. They have a huge group of roles in the immune system, and they are the precursors for osteoclasts. If you recall, in our bone lecture, we talked about osteoblasts are created from the bone stem cells, but osteoclasts are produced from blood. Well, here is the blood cell that produces them. It is the monocyte that differentiates into the osteoclasts. Finally, our lymphocytes. We'll deal with more detail of lymphocyte function in the lymphatic system. They're our major immune response. We call this our adaptive immune response, and they create our B and T cells. Okay, so that is blood. Make sure you understand the 5 leukocytes, general function, and you'll need to recognize them histology with the pictures I've given you. And then make sure you understand the development of blood, their stem cell precursors, and general function of those cells. Let's move on now to the heart. Now, the heart is actually one of the first organs that we're going to discuss that is found in the ventral body cavity. Specifically, this is found in the thoracic cavity, and there are 2 parts of the thoracic cavity. There's the pleural cavity, which holds our lungs. Then there's the mediastinum. Now, the mediastinum has an upper part and a lower part. The upper mediastinum is where the major blood vessels come out. The lower mediastinum is a separate cavity called the pericardium. Now, inside the pericardium is the heart. And if you recall, all organs found in ventral body cavities are surrounded by a serous membrane. And the serous membranes are named for the cavity in which they're in. So, for instance, the lungs are in the pleural cavity. Therefore, the serous membrane that surrounds them is called a pleural membrane. Since the heart is in the pericardial, or the pericardium, the serous membrane is called the pericardial membrane. And all serous membranes have two layers. We have a visceral and a parietal. So we call this the visceral pericardium and parietal pericardium. So if this was my heart, you two one day might be able to draw this heart. The visceral side is physically attached to the heart wall. Then it folds around on itself and reattaches. So this is the visceral side, this is the parietal side. So the visceral side attaches to organs, and the parietal side attaches to the outside, whether it be a body wall or maybe another organ. So here is a superior view. We actually see here's the heart. The visceral pericardium is right there. It wraps around the heart, folds back on itself, and it's actually now attaching to the parietal pericardium is now attaching to the parietal pleura of the lungs, and it attaches to the body wall. And then it reconnects on the other side. And so it creates a little sac, and that sac has fluid. We call this the pericardial sac with fluid, with pericardial fluid. One of the advantages of having a serous membrane surrounding each organ is it isolates each organ. It ends up being a physical barrier, so anything that's happening here can't just naturally diffuse over here. There's a physical barrier now. And with the fluid, we are decreasing friction. So as things are moving around, as our heart is beating or our lungs are expanding, we have some wiggle room. So we're decreasing friction by having that fluid. Now, each serous membrane has something unique. Let's talk about the uniqueness of the pericardium. When we say pericardium, we're referring to the serous membrane of the heart. So the pericardium actually has another layer. We call this a fibrous pericardium. Okay, this is just connective tissue. And this anchors and holds the heart and its blood vessels. So we have the pericardium up on top, holding the major blood vessels, and we have the pericardium on the bottom, anchoring the heart to the diaphragm. So if this was my heart, here's my diaphragm, I would have dense connective tissue anchoring it, holding it into place. That is the fibrous pericardium. Okay. And then I would also have some up here as well to anchor and hold the base. So this is the apex of the heart, this is the base of the heart, and coming off the base are all of these major blood vessels, which we will outline. Okay. Now, physically attached to the fibrous pericardium is the parietal pericardium, and this is continuous with the visceral pericardium. Now, visceral pericardium also has a different name. We call this the epicardium. That is, the visceral pericardium is the actual first layer of the heart wall. Underneath the epicardium, we have a myocardium, and this is our cardiac muscle. And underneath that is the endocardium. And this is our simple squamous epithelium. So we have the inside of the heart, its lumen. And we have learned in Unit 2 that epithelial tissue lines the lumens of organs. So this endocardial layer is considered simple squamous. Attached to that is our myocardium. Attached to that, remember, is our serous membrane, the visceral pericardium, but we call it the epicardium, excuse me, referring to the wall of the heart. And now that's physically connected to the parietal pericardium. So our heart is physically attached to our serous membrane. Okay? Remember what the epithelium is of the serous membranes. If you remember, we call it the mesothelium, but this is also simple squamose. So we have simple squamose, cardiac muscle, simple squamose, physically attached to the pericardial membrane. So our heart is not going anywhere. Let's look now again at cardiac muscle. Now, if you recall, cardiac muscle is different in skeletal muscle by leaps and bounds. Number one, it is an involuntary muscle. So we want it to work as a functional whole. Now, cardiac muscle is striated, and those are due to the myofibrils. So myofibrils, just like in skeletal muscle, are the long chains of sarcomeres with the I and A band, hence the striations. Remember, if you recall, these are short branched fibers with single nuclei, and they're physically connected to other cells by these protein connections called intercalated discs. So we still see the striations in them. Now, so another difference between them is when you look at a series or stacks of skeletal muscle fibers, they're going to have gap junctions in them. And like gap junctions of epithelial tissue, it allows for the cytoplasm to be shared amongst the various cells. So here are the gap junctions. And this is especially important in making sure that the muscles are all contracting as a whole. So the intercalated discs fuse those cells together. So when one muscle contracts, we're physically connected to the next. When one muscle fiber, one cell contracts, we're physically connected to the next cell. And the release of the calcium from their ER, it's the same mechanism for muscle contraction and skeletal muscle. When one cell releases its calcium, which causes activation of actin and myosin, that calcium can now diffuse into the next cell via gap junctions. This allows for all the muscles to contract as a whole. If you think back to skeletal muscle, right, they're broken down into motor units. So based off the force applied, you can use 5% of your muscle, 10% of your muscle, or all of your muscle. You don't want to really be playing around with 10% cardiac muscle. You want your muscles your heart muscle to contract all the time. That way we're maximizing the output, our cardiac output, the amount of blood coming out. Now we can speed up our heart rate, we can slow it down, but we never want to stop all of the muscle to contract. We want the full muscle to contract at all times. So by sharing cytoplasm and by having these cells fused together with intercalated discs, the muscle works as a functional whole. And then therefore there are going to be some pacemaker cells that set the stage for how the contraction works. And we'll discuss that during the cardiac cycle. Okay, let's now look at our heart. We have 4 chambers. We have the atrium on the right side and the ventricle on the right side. And then we have the atrium on the left side and the ventricles on the left side. Okay, so major blood vessels that brings blood back to the heart is the superior vena cava and the inferior vena cava. The superior vena cava and inferior vena cava are draining the blood. These are veins, so they're bringing blood back to the heart. General rule of thumb is veins bring blood to the heart. Arteries bring blood away from heart. So the veins are bringing blood to the heart from organs, where the blood is taking, or the arteries are taking blood away from the heart to organs. Okay. So the superior vena cava and inferior vena cava are bringing blood back from the upper body, the head and arms, from superior vena cava, and everything below the diaphragm for inferior vena cava. Now, that blood travels through the right atrium, out, down the right ventricle, and out an artery. We call this artery the pulmonary trunk, which leads to the pulmonary arteries. I'll put a little A there. This is traveling to the lungs. From the lungs, we're going to now bring the blood back to the left side of the heart. And these are called the pulmonary veins. As the heart enters the left side, from the left atrium it travels the left ventricle, heads out the left ventricle through the aorta. And this will now deliver blood back to the body. Now, it's very commonplace to think artery is red and vein is blue. But this isn't a vein. Here's our vein. I want you to think blue blood vessels, although yes, 95% of them will be blue, but blue blood vessels are blood vessels that have oxygen poor blood in them, and typically most veins do, except for one set of veins. And I want you to think, if you see red, you'll think oxygen rich, and most arteries carry oxygen rich blood. So if we're bringing blood back from the heart, here's the superior vena cava, we're bringing it to the right side. There's the right atrium, there's the right ventricle, and now we pump it out the pulmonary trunk. Well, this is blue. It's oxygen poor. However, it is delivering blood away from the heart to organs. It's going to the lungs. So it is an artery, but it's carrying oxygen poor blood, and that's why it's blue. From the pulmonary trunk, we branch the left and right pulmonary arteries. This takes our blood to the lungs. The lungs are able to get rid of carbon dioxide, which is metabolic waste from the cells, and bring in more oxygen. The pulmonary veins now come in the left side, and here is a back view of the left atrium. Notice it's red, because this is now oxygen-rich blood. From the left atrium, it travels down the left ventricle, and then gets pushed out the aorta. We have the ascending aorta, the aortic arch, and the descending aorta. The descending aorta is going to travel through the thoracic cavity, penetrate the diaphragm, through the abdomen, and then branch off to go to your legs. In this unit, you'll be learning blood vessels that go in and out of the heart, that go up to our head and down our arms. Part 2 of blood vessels will be in Unit 4, which will outline in detail the blood vessels of the lower body, below the diaphragm. So coming off the aorta, you end up having 3 branches. First branch is brachiocephalic. Second branch is left common carotid. And third branch is left subclavian. Let's think about that word brachiocephalic. Brachio refers to arm. Cephalic refers to head. So brachiocephalic is the blood vessel that will branch off and create the blood vessels for the right side of the head and the left arm. The right arm. The left common carotid will serve the left side of the head, and the left subclavian will serve the left arm. So these are the major blood vessels that you'll be learning, and their various branches heading up into the head and going down the arm. And that will be for this unit. Now, physically attaching the aortic arch to the pulmonary trunk is a little ligament here called the ligamentum arteriosum, our Harry Potter ligament. And this is a leftover structure. We call this a vestigial structure of the fetal circulation. So I will be outlining what that is in the blood vessel chapter. That blood vessel chapter will be covered in Unit 4. Okay. So let's look now on the inside. Okay. I'm going to redraw this because every chamber is closed off by valves. I'm just going to use a general atrium and a general ventricle because they're roughly the same thing. Okay. Within each ventricle are extensions of the myocardium. We call these papillary muscles. Okay. Now, the valve that brings blood from atrium to ventricles are called the AV valves. And there are 2 of them. On the right-hand side is the mitral valve. Is the tricuspid. So right-hand side is tricuspid. On the left-hand side is the bicuspid. The bicuspid can also be referred to as the mitral valve. So here is our first AV valve, the tricuspid. On the left side is our bicuspid. If we remember the order of flow, so right side first and then left side second, then a easy way to remember is tri before you bi. Tricuspid, bicuspid. Now, the doorways, if you will, of these valves are called leaflets, also called cusps. And they're physically attached to these papillary muscles through a structure called the chordae tendineae. Okay, here's those little cords. Now, these valves are meant to be one way. So, when blood flows through them, when the ventricles contract, we want this valve to close and force the next valves open. The valves leading from out of the ventricles are called the semilunar valves. And that's because of their shape. They look like half moons, so semilunar. They are named based off of the blood vessel in which they're coming out of. So on the right side, it leads the pulmonary trunk. So we call this the pulmonic valve. On the left side, it leads to the aorta. So we call it the aortic valve. Okay. So don't just say semilunar, because the question will be which. Or don't just say AV valve, because the question will be which. So we have the tricuspid, which leads us into the right ventricle, and then the pulmonic valve pushing blood out the right ventricle into the pulmonary trunk. Blood returning from the lungs enters the left atrium, heads through the bicuspid valve or mitral valve that leads us into the left ventricle, and then we get pushed out the aortic valve to go through the aorta. Okay. All of these valves open and close based off of pressure. Okay. So, when the muscle contracts, that is, when the ventricle contracts, right? So, let's say muscle contraction. That's going to lead to an increase in pressure. This is blood pressure. And that pressure is going to push out in all directions, including wanting to push up the AV valves and cause blood to backflow. Well, when muscles are contracting, so are these papillary muscles. And so they're pulling down. Think about the direction that the chordae tendineae are going to pull. They're going to pull down as well, which seems counterintuitive. But look at the direction of the force. The force is going in the opposite direction as the cords are pulling. Let's simplify this. Imagine we have two chambers, and they're separated by a doorway, in this case the valve. Now, if we were to put pressure on this, right, the valve would open and our blood would travel through. If we were to put pressure on this way, our valve opens the opposite way and blood travels through. But if you were to apply a force in both directions, the valve stays closed. So in this case, opens. This case, opens. Right? This case, closed. Now, imagine that we take this force. See if I can. And apply it to the other side. So now I have a force pulling the door open in one direction and a force pulling the door open in the opposite direction. That's exactly what we see here. When the ventricles contract, we want to push these valves open, and that would cause backflow. We don't want that to happen. The chordae tendineae are pulling in the opposite direction. Opposite direction. So no matter how hard the ventricles contract, that contraction is tied to the muscle. And part of the valve is a papillary muscle, right here. So as the papillary muscle is contracting, we're forcing blood up into the AV valve, and this ends up shutting the valve. It is unable to prolapse and push blood back into the atrium because the chordae tendineae are pulling with an opposite force. So we're kind of like this here. We have an opposite force pulling in one direction, and the pressure is forcing the opposite direction. and therefore we stay closed. Now, for the semilunar valves, it's just a matter of pushing enough force for the semilunar for it to open up. And that has to do a lot with how much blood is left over, because any blood in the arteries are going to put a force against these valves, the semilunar valves. So as long as my contraction of my ventricles can force these valves open, I'm guaranteeing these valves are closed. So all that force building up is going to force those valves open as soon as the pressure in the arteries go away. And the pressure in the arteries are going away because the blood is being delivered to organs. So eventually this pressure in the arteries diminishes and the ventricles have enough force to open them up. So all of these valves work on pressure, and that pressure is created by the muscle contracting. We contract the muscle, it increases the pressure, and the pressure pushes out in all directions. The chordae tendineae guarantee the AV valves are going to shut and not prolapse. Therefore, we're prohibiting being able to be pushing our blood back. Therefore, the only exit route is the semilunar valves. And with enough force, we can open them up and we can go out. Now, the way the semilunar valves are open, the way they close with these little half cusps, and they're unable to open the opposite direction. So where the AV valves are technically two-way doors, we need these extra structures to guarantee it's a one-way flow. But the connective tissue network of the semilunar valves are set up to where they can't backflow. The reason why I just drew one atrium and one ventricle is because they work together. The atriums contract at the same time, pushing blood in from atrium to ventricles, and the ventricles contract at the same time, pushing blood out. So when the AV valves are open, we're filling the ventricles, therefore we want to keep the semilunar closed. When we're ready to contract the ventricles, we're going to close the AV valves and therefore force open the semilunar. It's the closing of the valves that make our heart beat. So that bump, 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 bump sound you're hearing has to do with the physical hitting of the valves closing. So think about a snap. Right? That snap is due to your thumb or your middle finger hitting the pad of your thumb. Okay? So that bump, bump, the first bump is the AV valve closing, right? That open, then the semilunar open. The second bump is the semilunar closing, and then the opening of the AV valves. They call it lub-dub. I think that's a dumb name, but it's lub-dub. I just call it bump-bump because I hate lub-dub, but it is what it is. Okay? All of these openings or closing are based off of pressure, and it's the pressure in the ventricles. The pictures in your book make it seem as if the atriums and the two ventricles are roughly the same size. 80% of the heart is the left side. It has a nice thick muscle. That's because it's contracting and pushing blood out to the entire body. The right side is a little bit thinner. It doesn't need to be as large. In fact, if we look at it, look at the thinness of the myocardium on the right side versus how thick it is on the left side. The right side is only pushing to the lungs, which are literally next door, about 30 centimeters. The atriums don't really need contraction. They do contract, but you can kind of think of it as a topping off point, because as the blood enters, just by gravity, it's going to pull into the ventricles. So our atriums contract as more of a topping off point, and it's just a process of getting the electrical current to, after we contract the atriums, to then travel down and contract the ventricles. So it really is about ventricular contraction and less about atrial contraction, but they do happen at the same time. Okay. Now, the cardiac muscle is considered involuntary, but we have some pacemaker cells called nodal cells. And they set the contraction pace. That's why they're called pacemaker cells. The first group of pacemaker cells are called the SA node. Okay. SA stands for sinoatrial. We find this up in the right atrial wall. And it's going to send signals down to another group of nodes called the AV node. Okay. This is kind of buried in the right atrial wall a little bit lower. So this is upper wall. This is a little bit lower. So right atrial wall, kind of between the ventricles. So I'll just put lower. There is a series of pathways that deliver the electrical current from the SA node to the A node, the AV node. We call these internodes. And so when the nodal cells send electrical impulse down, that impulse carries down to the AV node and activates the AV node. Now the AV node has bundles heading in between the two ventricles. Okay, we call these the bundle branches. These head down the bottom of the ventricles and create what we call the Purkinje fibers. So these are the very, very bottom of the ventricles. And it's the Purkinje fibers that will then contract and push the blood from bottom up to the ventricles. So our SA node causes, when it activates, it causes contraction of the atrium. So atrial contraction. That current is sent down to the AV node. And the AV node sends the current down the bundle branches, up through the Purkinje fibers, and that causes ventricle contraction. Now this is all on the right-hand side. Coming off of the SA node is a group of bundles called Bachmann's bundles. And this takes us to the left atrium. So we have another group of bundles here. It doesn't show it, but even though the SA node is in the right side, these internodes take us to the AV node. The bundle branches move us to the left side. So the left and right atrium contract at the same time. At this end, as the AV node gets activated, it sends signals down the wall between the ventricles. This is called the ventricular septum. And it heads it up the Purkinje fibers and causes contraction of both ventricles at the same time. Now there's an issue of spacing and timing of this pathway. For some reason, one of the papillary muscles is a little bit further away on the right side. And so if this was the only path that I drew, then one papillary muscle on the right valve, that's the tricuspid valve, would contract at a slight second behind the rest of the contraction, which would make the AV valve not shut as thoroughly. So we have a shortcut, and the shortcut takes us to a papillary muscle, This shortcut is called the moderator band. And it's actually an extension of the heart wall layer that takes another branch to a papillary muscle. Let's go back and look. We can actually see inside the heart. The inside of the heart has a network of tissue. It's not flat. It's just this huge network, like branches. They're actually called trabeculae carnae. And one of these branches right here is the moderator band. It's carrying the nodal signal to one of our papillary muscles. So that way all the papillary muscles can contract at the exact same time. So the moderator band is a shortcut. So as the bundle branches head down, one splits off and goes to the moderator band, through the moderator band to one of the papillary muscles. The rest of the pathways head up as Purkinje fibers along the walls of both ventricles. So it's a unique structural feature that we see in the right ventricle and not the left ventricle. Okay. This creates what we call the cardiac cycle. There's a couple new terms I want you to be aware of. We have systole and diastole. Systole means to contract, and diastole refers to rest. So, the AV valves are open, blood is returning, blood can freely enter into the ventricles. When the AV valves are open, the semilunar are always closed. The reason why is there's low blood pressure. Right? There's no pressure in the ventricles because they're at rest. So we call this ventricular diastole. And any blood in the arteries will close the semilunar because once again it's all pressure based. So you put some blood in here, you slam it shut. There's nothing forcing that valve to be open. Now, the SA node contracts. It activates atrial systole. A little topping off. That starts the cycle. Atrial systole. We don't want both atriums and ventricles to contract at the same time. It wouldn't go anywhere. So as blood is pouring into the ventricles, we have atrial systole that tops off the ventricles, and now the atriums go into diastole and they rest. And now we lead into ventricular systole. So now the AV node gets activated. It heads down the bundle branches through the Purkinje fibers and moderator band. It contracts, and now we begin to build pressure. That pressure slams the AV valve shut, and the papillary muscles prevent from prolapsing and pushing it back out. Meanwhile, we're trying to beat the force of our semilunar valves. And this takes a little bit of time, but eventually we build up enough pressure to open them up. Now we allow the blood to leave the ventricles, which now decrease the pressure, and we go at rest again. So now we're back at ventricular diastole. As the blood is returning, enough force will open up the AV valves again, and then we start the cycle over. So we have contraction, contraction, followed by rest. One contraction will so we'll have the closing of the semilunar valves once we rest, then the opening and closing of the AV valves once the ventricles start to contract. So contract, atriums, contract, ventricles, rest. Contract, contract, rest. Contract, contract, rest. Contract, contract, rest. So those contractions are increasing blood pressure, allowing those valves to close one and open the other, hence giving us our heartbeat. In physiology, you'll learn about isovolumetric contraction and the P waves and QRS complex and all that fun stuff about the EKG heart rhythm, I wanted to focus more on how the structural elements of the heart open and close based off of these contractions. Everything else you will learn in physio. Okay, that is the end of both Chapter 20, Blood, and Chapter 22, the Heart. If you have any questions, please let me know.